Hi, everyone. I want to welcome you to our Sarcoma Sisters podcast. And we're here today with one of our friends who is an expert in pediatric oncology and also an expert in pediatric genetics and genomics and all that molecular testing that we talk about nowadays. We want to welcome Dr. Brian Crompton to the show. Woo!
Hi, everybody. Thanks for having me.
He's a sarcoma brother. You forgot that part.
That's
Yes.
right.
And of
I'm a
course,
sarcoma brother.
he is the brother to our sisters. So yes, we're so happy to have our sarcoma brother. We've been talking about it for a while, and we're so grateful to have you here today. So, September, it's so great to have you on September because it's Childhood Cancer Awareness Month. And we're so happy that you have your cool background that everybody who's watching us on YouTube will see. But we just want to
And my
start
yellow
out
shirt.
with,
It's a gold shirt.
oh yes, I know you had the
Go
gold
gold.
shirt too. Go gold, go gold. So to start us off, we just wanted to ask you what led you into pediatric oncology and taking care of pediatric sarcomas?
Yeah, I mean, I get that question a fair amount from people, especially, foundations and things. They're asking, why did you go into this? And I wish I had like a really amazing story. I think the reality is you have to have a very similar maturity level to your patients. And so adult medicine just isn't going to work out for me. So that's how I ended up in pediatrics. No, I love kids. And it's really, I don't know, it's just so much fun working with kids. When I was a pediatric resident, a trainee, I was really interested in going into a field where you could really balance both taking care of patients and doing, really hardcore research. I was really interested in lab work. And so, as I was looking around for a field where, that all that came together, there's actually not that many options. And, pediatric oncologists there's a long tradition of doing patient care and research. And that research, really has fundamentally changed how outcomes are for pediatric cancer in the last couple of decades. And we're not really like, I was also at the time thinking, well, what's going to also sustain a 40-year career of sort of making gains in the field of medicine? And we're not close to the finish line, right? I mean, that's part of why Childhood Cancer Awareness Month is so important.
Right.
We cure maybe about 70% of kids with pediatric cancer, but to do that, we have to give them incredibly toxic therapy. There's all kinds of long-term side effects. And 30% of patients not being cured is an enormous number when you think about it being the leading cause of disease-related death in the United States. So between being a big giant child, wanting to do really like fundamental laboratory research, but still have a really important, deep connection and relationship with patients, this was the right fit. And then when I tried it out for the first time, I just like totally fell in love with the field.
Thanks for sharing that. Yeah, that's awesome. And especially for all the information on why it's so important and why we need to raise awareness for childhood cancer, that even though the cure rate might sound good compared to adult medicine, we still have so far to go. And there's so many long-term late effects.
Yeah.
But then that leads me to my next question. So like you said, you do work a lot in the lab and you have your own laboratory at Dana-Farber. Can you tell us what you do in the lab?
Yeah, so we're specifically focused on sarcomas. Our work really started out in pediatric sarcomas, of course, as a pediatric oncologist, but we've now started multiple collaborations sort of across the spectrum with some adult oncologists who work in sarcoma research. And my lab is very translational. So we're trying to leverage laboratory techniques to improve outcome. And some of that is oriented around trying to discover new ways to target sarcomas that could potentially lead to new therapies. And that's definitely an important aspect. Another part is to really use genomics as a way to both understand the biology of these tumors and also really impact the way we're treating patients. And so one example I like to give is that in leukemia, where patients have cancer of the bone marrow. Bone marrow is that spongy area inside the bone. That's where leukemia arises. It's very easy for clinicians to sort of check the bone marrow to see how it's doing along the way during a patient's treatment, and that's exactly what happens in leukemia therapy. So they can do what's called a bone marrow biopsy, and they get a sample of that bone marrow. It's not very painful. It's very simple to do. It's not dangerous. They can look at it under the microscope. They can sequence it. They can do all kinds of tests. And then after just a couple of weeks of therapy, they can look at it again and say, how's the patient responding to therapy? And they started doing that maybe 15 years ago, and it completely changed how patients with leukemia are treated, because they were able to identify very early on patients who were not responding to what is considered standard of care therapy. And so they intensified therapy for those patients where their bone marrow didn't seem to be responding. And those patients now do just as well as patients who were responding well to standard therapy because they've intensified therapy. In solid tumors, we can't do that at all in sarcomas specifically, because we get maybe a teeny fragment of tumor biopsy sample at diagnosis. Most of that's used to sort of say what the diagnosis is, what subgroup of sarcoma it is, and then we almost never sample the tumor again. So we have no idea how patients are responding. And we know, especially like in bone sarcomas so sarcomas that arise in the bone, even tumors that respond on imaging, you can't tell, because where there was that big mass in the bone, when you go do imaging again, it still looks like a big mass. It's just some of it hopefully is dead. And so imaging has been really ineffective for us in terms of measuring those same type of responses. And it's really just completely precluded us from being able to change therapy based on how well patients are responding. And if you think about it, that is really sort of like the ultimate in personalized medicine. Like if you can figure out which patients are responding, then you can change therapy for that patient, then you really are doing personalized medicine. And so we're trying to use genomics to kind of create brand new tests that don't require multiple surgeries, so that we can start to measure patients' response to therapy and maybe change the way we do treatment forever for patients with sarcomas.
Can I interrupt you for a question?
Yeah.
Brian? Can you give me your 2-sentence elevator description of genomics?
Yeah. So 2 sentences. That's very short.
I should have prepped you for the question, but I think
that's okay.
I think people hear precision medicine, genomics, molecular genomics, genetics. It all sounds like blah 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 blah. So what is genomics?
Well, I think the fundamental part to understand is that what makes a cancer cell primarily different from a regular cell is that it's acquired some mutations. And mutations are changes to the DNA sequence. Every cell in our body has DNA. That DNA tells the body how to act. It tells the cell how to act. It tells it whether to be a kidney cell or a lung cell. But when that DNA sequence, the pattern of bases that make up that DNA change is called a mutation. And that mutation totally messes up the programming. And so genomics is a way to look at cancer cells or normal cells and sort of see what is the complete list of mutations that cell carries that could be contributing to why that cell has now become a diseased cell, in this case, a sarcoma cell. And so when we talk about genomics, we often talk about taking a piece of a tumor and profiling it or sequencing it to find all the mutations that make it a cancer cell now, and not just a normal muscle cell or bone cell, for example. And then the idea of precision medicine is that if you can find the mutations that cause it to be a cancer cell, and those mutations cause cells to be vulnerable to a very specific therapy, then you can use those genomics, those mutations, to find treatments that will specifically kill that person's cancer cell. And that's what's meant by precision medicine or personalized medicine, is that if we can understand your tumor very well, we might be able to give you therapy that's specific to your tumor and not just specific for everybody like you who has the disease. And we know everybody's tumor is different. So the closer we can get to understanding your specific tumor, the closer we might get closer to giving you exactly the therapy you need to be cured from your cancer.
Thank you.
That was way more than 2 sentences.
That was more like a long walk to Starbucks, but I'll take it.
Sorry, I don't know how to do that in 2 sentences.
So you said that, we know for leukemia, oopsies, I'm making a mess. we know for leukemia that we can sample those tumor cells, which are leukemia cells, and kind of check on them, see how they're doing. We don't do that for sarcoma. And we talked with Dr. Hayes Jordan a couple weeks ago about sarcoma surgery. And I'm just going to interject this, and then I'm going to ask the next question. The reason that we often don't do that for sarcomas is because those cells are hard to get at. They're in the bone, they're hiding in the abdomen, et cetera, et cetera. So what are you doing in your lab right now to try to make sarcoma cells and sarcoma genomics more accessible so that we can study these easier without major surgeries?
Yeah, so maybe about 10 years ago, maybe a little less than that, this brand new field of oncology research opened up, sort of out of the blue, and an extension of how good we're getting at doing genomics. It turns out that as tumors grow, there's a certain amount of that tumor that maybe is outgrowing its blood supply or just gets too big in certain places. And some of those cells within the tumor, even though most of the tumor is growing, some of the cells die and they release material when they die, and that gets into your bloodstream. So it turns out if you look, you can find DNA from tumor in patients with cancer in their blood. And so if you know where to look and you have really sensitive genomic tools, you can detect the presence of what we call circulating tumor DNA in the blood. And so what's amazing about it is that the amount of this, sort of free-floating tumor DNA that's in the blood actually correlates with how much disease you have. And as patients go on to therapy, the amount of this circulating tumor DNA can go from high, and if they're responding well, can drop really low. And that can be a measure of response to therapy, as one example. It also turns out that our sequencing technologies are getting so good that we now need very little DNA material to do that sequencing we talked about, that genomic profiling we talked about. So just from a blood sample now, we can basically profile a tumor from a blood sample and see all the mutations that make up that tumor by doing kind of comprehensive genomic profiling. And then what's even more interesting, I think, is that if there's a patient who has multiple sites of disease, usually our biopsies go to one spot and we sequence that one spot. But we know that cancer is sort of like this, natural selection or evolving beast, where as time goes by, it acquires new mutations and changes. And that biopsy can't capture that because we're looking at one site where we know that if patients have metastatic disease, where disease is spread to another site, that those metastatic sites may be acquiring new mutations that aren't present in the primary site that got biopsied. So what's interesting about liquid biopsy is it turns out studies show that all those sites likely shed tumor DNA into the blood. So when you profile the blood, you get sequencing from all those sites. It's all mixed together, but you can see all the variants that might be contributing in all different parts of the body. And so I think, we're still in the early days of liquid biopsy, especially for sarcomas and pediatric cancers, which, rare diseases just take more time to develop technologies for. But I think we may, down the road, get to the point where we find that actually doing a profiling of tumors from a blood sample is actually more informative than doing it from a biopsy that happens only in one spot of one tumor. And so that's what my lab works on, is trying to adapt those liquid biopsy technologies, most of which have been developed for things that are very common, like breast cancer or lung cancer, and trying to adapt them to sarcomas, pediatric sarcomas, adult sarcomas, and other rare pediatric cancers as well, so that we're not falling behind, and we can take advantage of these technologies to understand the biology of these cancers better.
Thank you. I think sometimes when I've talked to patients about, the tumors kind of shedding this DNA and stuff, it sounds crazy, right?
It does sound crazy.
But I found that one good way to explain this, and I just wanted to say this for our listeners and viewers, is I think a lot of people who have had children recently in the last 5 or 6 years, we now know that you can get a blood test and see if you have a single pregnancy, not twins. you can see what the sex of your baby is, your biologic sex of your baby, because that baby is doing the same thing. It's releasing
That's
that
right.
DNA into the blood. So that's essentially the same process, right? A little bit different, but it's the same technology.
Yeah, I'll take it a step further. Cancer research completely stole the idea from fetal medicine. It's absolutely true.
Really?
That the
That's so cool.
obstetricians and the fetal medicine scientists came up with this idea first, that you could find, if you had a woman with pregnancy, that you could find traces of the fetal DNA, and you can do all kinds of profiling now and understand the genomics, the normal DNA of the baby before the baby's born from a mother's blood test. So of course that instantly made cancer docs think, well, why shouldn't we be able to find tumor DNA there too? And of course it turns out to be true that we could do it. So you're right, it's not crazy. It wasn't even our idea originally, and it works really well.
It works smarter, not harder, right?
That's right.
So the next thing I wanted to ask you, so you said multiple times throughout there that in the future, down the road, we're getting good at this.
Yeah.
So can you talk to us about what that means for patients right now that, say, is diagnosed with rhabdomyosarcoma, and we get multiple time points and we say, this is the profile of your tumor. What can we do with that information now versus what do we hope to do with it in the future? And why is it important that we get that information now from not only that patient, but other patients?
Right. So, I mean, I think one of the things that has to be incredibly frustrating for families and patients, and is incredibly frustrating for me, is that before you can use anything in the clinic, you really have to prove that it works, that it does what it says it's going to do. And I think with liquid biopsy, we're still very much in that phase where we've at this point proven scientifically that we can detect the presence of it. And in a research lab, we're able to profile it, profile the ctDNA, and in that sense, profile the patient's tumor. But most of that has not yet reached sort of the point where we can apply to like the FDA for approval as a diagnostic test, for example, or as a return of results kind of test. There are definitely companies out there who are doing this. And so I don't want to necessarily say companies' names and bias anything, but there are companies where you can pay to have a blood sample sent off and they will return a profile of the patient's tumor from that blood test. And I think that we're still trying to figure out at this stage whether those tests are good enough for our purposes for pediatrics and sarcomas. I think for some things that we want to do, it will work. In other aspects, those tests are not ideal for the types of tumors we see. And I can explain a little bit if you want about why that is, but it really has to do with what the mutations are that make sarcomas different from carcinomas. All those tests have been developed to be optimized to detect DNA from carcinomas, not from sarcomas. And so I think part of what we need is to figure out exactly what tests are good for what. And the way we do that is by collecting samples prospectively on every trial we do in sarcoma. Every institution can run a prospective trial in breast cancer because there's a lot of patients with breast cancer. For sarcomas, we really need to use every multi-institutional trial because these diseases are rare enough to collect every sample we possibly can. And that's going to allow us to sort of really validate these assays and say, this is what works. These are the types of liquid biopsy tests that work for this clinical utility, and these work for that utility. And in some cases, if we have to make our own and we validated our own, we can go to the FDA or go to an industry partner and create a commercial test that will work where the existing tests won't work for our patients.
Yeah, I think that's a timely conversation in the world of the COVID-19 pandemic, where we've seen shifting results of the sensitivity and specificity or the accuracy of a certain diagnostic test, and how it's really important to get those things right. And obviously that's a different scenario. We're under a public health crisis at this moment, but getting the right answer is as important, if not more important, than getting the answer in a certain time frame.
Yeah.
And so if a patient can't, if we don't have the capability right now to use this clinically, why do you feel it's important to develop this in parallel? So I guess let me rephrase my question. So if I have a patient who comes in who has a sarcoma and they look like they're not responding to therapy, I have a limited number of options of what I can switch them to, right? And there's a whole different set of researchers that are working on new drug development, you know, immunotherapy, those sorts of things. So can you talk about how those people that are looking at new therapies and developing new therapies can utilize what you're developing in parallel, and why we have to develop new therapies and companion diagnostics or these kind of blood tests in parallel so that one doesn't outpace the other, and so we can use them at the same time in the future?
Yeah, I mean, I think what's so complicated about all of these, you know, if we just sort of put ctDNA under a bigger umbrella of companion diagnostics or companion tests, what's so tricky about doing that is the test has to work in the context of the exact drug you're trying. So if you have a patient who goes onto an early phase trial, it may be that ctDNA levels, as an example, drop precipitously on chemotherapy, and that correlates with response, but that may not turn out to be true for a targeted therapy. Maybe ctDNA levels will go up and patients are responding. So basically we have to revalidate it in the context of that drug and that therapeutic approach. So that means we shouldn't wait until the drug's approved, because we don't want to have a drug and then not know whether patients are responding to it. So every time there's an early phase trial, especially one that I hear about or one of my friends is running, I raise my hand and say, can we please collect samples? Everybody's laughing because they know this about me. I say, can we please collect serial blood samples on this study, because we need to validate in the context of your drug. And if that drug doesn't work, we spend a lot of time validating something that, you know, may not even go become a real drug. But if it does work, we're not like way miles behind. And so, you know, one of the things people laugh at me for, because I'm so obnoxious about it and I deserve to be laughed at for, it's just every time we do a trial, I like really hound the investigator to say, what kind of biology can we do? The liquid biopsy is obviously one area, but getting tumor tissue whenever there's a biopsy is another. Making sure that like other markers of response are being under consideration, that we have all the best scientists who think about that field and that particular mechanism of drug activity involved in that clinical trial, so that if a trial works, we really understand why it worked. And even perhaps more importantly, if a drug fails, why did it fail? Like, you know, we don't try drugs unless we have some scientific evidence from the lab that it probably is going to work. And yet most of our drugs don't work, and we never learn anything out of those failures. And I think for the first time with liquid biopsy and all the other new technologies that are coming down the pike, we may finally understand why sometimes drugs that look really promising don't work. And why that's important is that allows us to then say, well, maybe that drug didn't work not because it stinks, but because it needed also X. And so if we put this drug together with that drug, now we've got a blockbuster combination that's going to cure people. And right now what happens without having any of that correlative biology is what we call it, any of that correlative biology built into trials, what ends up happening is that drug doesn't work and the community moves on. And we never learn anything from it. And that to me is just sort of a massive tragedy. And I think that we're at a place where we can really change that. And, you know, the truth is, it's actually quite easy. You know, clinicians want partners who want to do biology. And they don't necessarily have labs, so they can't always do it themselves. But if we have a group of translational researchers like myself, like you, Wendy, and others who really want to do these kind of things, it's actually quite easy to convince our clinician colleagues or the trialists to let us do these studies. They're more than happy to help because they want to learn from their successes and failures as much as we do, as much as the patients want us to.
Lisa, I did it again. I'll let you ask your questions. Sorry.
Oh, no, you're good. I was just going to reiterate and to say, in case anyone wasn't aware, that early phase trials just means that it's like either a phase 1 or a phase 2 trial, meaning that the drug has been tested in the mice and we've cured lots of mice, but now we want to test it in our patients and see if it, what the safety is of the drug, but then if it has any effects too. And Brian, I just think what you said, of course, you're hitting the nail right on the head. It's so important because I think that is really frustrating for families when it's either themselves or their loved one, they have a sarcoma that has come back and they decide to really give to future patients by enrolling on a clinical trial. And then they find out that the drug didn't work, even though it was never, it's not that it's guaranteed. The physician explains that it might not work, but then also that, well, then it was just scrapped. And, I was involved on an osteosarcoma trial where it ended up not working, the drug, but there was a couple patients that did respond. And that was kind of frustrating too, because it was like exactly what you're saying. Well, why did those patients, why did it work well on their tumor and not the other osteosarcoma tumors? And just you stressing that getting that tissue is just as important as developing new drugs, because if we develop a bunch of new drugs and they don't work for anyone, then we're still not getting ahead. We're just, like you said, it's like it's a shame, because then we're just wasting, it almost seems like wasting time. And I don't like to put in that context, but that you have all these fancy new pants drugs and then we're not getting any responses from it. So I think that's important. And just what you're saying, that patients are so generous to even enroll on these trials, but then also to check that box to allow their tissue to be given. And then if it's safe and them and their physician thinks it's safe to do, to have a biopsy sent, because you're going to be helping so many patients in the future by doing that. So it was more of a long-winded walk to Starbucks comment, Wendy.
Everybody's going to Starbucks these days. Yeah, no, I
don't even like Starbucks. It tastes burnt. Blah.
I think we're going to have to become unsarcoma sisters after that. Brian's probably
I like more fancy pants coffee.
Are you Dunkin' Donuts, Brian?
I'm not super picky. I like Starbucks coffee, but I tend to drink just like unsweetened, no cream or sugar, no sweetener coffee, either iced or hot. So really, it's really hard to screw that up, and so I'll go anywhere for it.
Excellent.
I drink a lot of coffee.
Anyways,
no, we could go back to this. And one thing I wanted to bring up that I think it's always really tough, and when I was taking care of patients too, is that you would get this genomic or molecular profiling done on the tumor, and it would say that certain drugs might work, and then you may get them or may not get them. And then a lot of the times they don't seem to work. And I just wanted to get maybe yours and Wendy's both thought on that about should we be doing those tests on our patients when they relapse, and what's the best way to go forward with that, because it is really disheartening, but then it's like you want to offer your patients everything that's available.
Lisa, are you specifically, I think we can probably call out a couple of the commercial tests by name, because I think in the oncology community, people know it now. But I think what you're referring to is when somebody relapses, sending in tissue to either like a Foundation One
or Caris. Yeah, because I've actually talked with Brian about this before, and this was a few years back when I was seeing patients, and I was saying, oh, the test says that we should use this, and Brian was like, well, actually, it's not that simple. So maybe the question is, why don't patients respond? Why isn't it that simple based on just what the output says? It looks like, this might work, even though the test is not saying that it will work 100% either. I have to specify that, but they are giving suggestions.
Well, it's a very complex problem that probably is a very long walk to Starbucks answer, but I think there's a couple of things patients should keep in mind. One is that these tests cost money, but if you take money out of the equation, which sometimes you can't, but if you could, I tend to be the kind of person that believes more information is better. So I participate in some national or multi-institutional studies that are specifically looking at genomic data, things that are generated similar to Foundation One. And one of these groups that I'm involved in that's run out of Dana-Farber by Katie Janeway, runs a multi-institutional study, and I'm part of her team. What we actually, her study actually accepts FoundationOne as one of the ways to get your tumor profiled. So I think, I tend to think that more information is better if the quality of the information is high quality. And that's kind of the kicker in some of this, is that when you're going to a commercial company, there's a couple of things. One is that they're trying to sell you a product that they need to show works. And so they're incentivized to give you a report back that not just says, here's your list of mutations, but here's some drugs that might work in that context. Because if they just report back, here's your list of mutations and nothing's going to work, that's not great marketing for them, right? So, and I don't want to demonize these companies, because I think actually a lot of the companies, they're companies that need to make money, but a lot of their medical advisory board are oncologists who really deeply believe that genomics can help improve outcome for patients. So in no way am I trying to demonize them, but there is a component of they need to find things that match what they're seeing in the mutations, because that's what they're promising to do. Mm-hmm. Also, the other piece of it is that, again, most tests are developed for common diseases, right? And so a lot of these tests that FoundationOne and other groups are sending or have developed are really optimized for to find the types of mutations you typically see in carcinomas, the common cancers, breast cancer, colon cancer, lung cancer, the things we mentioned before. So you can miss some important things in those assays that are important to pediatric cancers or sarcomas. And then the last thing, I think the third most important thing, is that if you find something in these tumors, you have to understand them through the prism of what's available for the patient. And the people who really understand what drugs are out there where your patient is eligible for are physicians who take care of those types of patients. So one of the things I love about Dr. Janeway's study is that where the sequencing happens, what company does it or what academic institution does it, actually doesn't matter as much as the real value of that multi-institutional trial is the expert panel she's assembled of pediatric genomic experts, not just genomic experts, but pediatric cancer genomic experts, including sarcoma experts, and clinical researchers and clinicians who specifically take care of those types of patients. And they understand what drugs are out there, what drugs their patients are eligible, because in kids with sarcomas, age often matters. So if you have a great drug that hits the target and you have to be 25 or older to get the drug, that recommendation is useless to you. So, and that's where I think that if you do these tests properly and give people the information that's really precisely useful to them, they can be really powerful tests. So I would just encourage listeners to go ahead, get those tests. But when you get the results, before you sort of dig into them yourselves and get too far ahead of yourself, meet with your oncologist, sit down and talk about what's possible. Maybe even have that conversation before you send off the test, so that you know what the likely potential outcomes could be of that. Because I think your doctors are going to know what tests, what types of trials you might be eligible for. And that's really the goal here of most of these tests, is that, the standard chemotherapy hasn't worked. You're looking for something new, and you're hoping that the sequencing tells you which clinical trial to try. And so I think if you have a doctor who's willing to sit with you and spend time with you, reach out to experts in the field, those tests can be really useful, and I encourage people to do them.
Yeah, I think, Brian and I come from similar institutions, but, before I was at Mayo Clinic, which is also a big institution, who has a good genomics program. I'm still getting to know the institution. We had big genomics programs, and so we had our own in-house stuff. And, I think one thing that's important to me as a clinician, coming from two heavily based research programs, is when patients come with these results, I feel a very deep sense of responsibility that even if that drug is FDA available, and even if I have a dose for it, and this is very relevant because this just came up, that I feel very strongly that to not only save the patient some money of the cost of drug, but also to learn from whether or not they do or do not respond to this drug that's been predicted to help. I feel very strongly that I really try to get them on a trial because I want to help them. And if I can't get them on a trial, I will do what I think is best for them always. But I owe it to not only them to help them feel better, but to my patients in the past who I've either not been able to help or have helped, and to my patients that I will have in the future, that if there's something that we can learn from them, that we capture that data. Because if I just do it and I have that information and it just lives up here in my head, that doesn't do anybody any good except for the next 5 patients that I see, but there's 5,000 patients that might need that information. And so, that's not available to everyone, depending on where you live in the country. Lisa, I grew up in North Dakota. There's resource limitations to certain geographic distributions, but I think that's something that I love about pediatric oncology, is that most of us are very like-minded in that fact that we really want to try to get information that we can from patients, and we're also grateful that they supply, that they allow us to enroll them on trials. They're usually very generous and gracious with, tissue submission, blood submission, saliva. And that, I think most of the pediatric oncologists in the United States, and probably in Europe too, really strongly believe in trials, not because we want to test our children or use them as test subjects, but because we want to help and we want to learn from them. We want to learn from their successes and we want to learn from their failures. So I think you're absolutely right. Those reports can be extraordinarily helpful, but you need somebody who can interpret it. And then, if there's a trial 40 minutes away, then I'm going to send my patient to that trial so we can learn something from them, and potentially save them some money too, because some of these drugs are real expensive.
I think clinical research and translational research, in some ways it's a contract, right? I mean, with the patients, that where there's a give and take, where of course we, just along with the patients, hope that something we're trying in an early phase trial is going to work, and that does happen sometimes, and that's the most important thing. But in return for them agreeing to participate or try a therapy in the context of a clinical trial, with all the obligations that go along with it of checkups and blood samples and all that, I really think we have an obligation to make the absolute most of learn the most possible we can out of every single clinical trial. And I think we now have tools to do that much better for solid tumors. And of course, that's what we want to do. I think we're capable of doing that now more than ever before.
Yeah, one thing I have been learning, I'm putting together a lecture on big data and oncology, and what's really exciting is that one thing in one of the papers I was reviewing was that, even if it's a small clinical trial with this term big data, you can actually, they've done studies where they've looked at all these small trials and then been able to have conclusions from them that you weren't able to see when it was just one trial at a time, because they didn't enroll that many patients. So even if it's available for a patient to be on a clinical trial that's maybe not big, or, it all makes a difference, because you're still collecting that data somewhere other than Wendy's head, even though her head is impressive.
It is impressive.
It all is all going to fall out eventually. Yeah, no, I think, clinical research, translational, which we've been throwing that word around, I'll just kind of shortly define it. We say bench research is like people playing with the cells and, doing the sequencing or whatever. And then translational is Brian, who's kind of bridging in between the clinic and the people at the bench doing that stuff. But it's so complex. And I think, we touched on this with Dr. Painter, that these rare diseases, which all of childhood cancer is a rare disease, and then when you break it down into the hundreds of subtypes, these are some really significant challenges that we run into. And I'm just so grateful that, I don't have a great story of why I became a pediatric oncologist either, Brian. So, don't worry about that. But I am grateful that, I found my people, right? The people that care, the people that will take an hour out of their day to do, a podcast to talk about all the intricacies of research. And so I think it's just so incredible to be a part of a community that's passionate, literally from developmental biology all the way through to, phase 3 and epigenetics and epidemiologic studies and all of the things that, you know, it takes this whole village to do, and everybody's so passionate about it and really trying to just row in the same direction, whatever your area of expertise is. So
Yeah.
Brian's our ctDNA guy. I'm grateful for that. And
Yes.
and I'm so sad that we can't get together in next, is it next week? Next week is our
Next week is Children's Oncology Group. Yeah, it's been a
Where we
weird
usually
year.
all see each other.
No.
We'll virtually get together, but yeah, this I think is just scratching the surface of all of the kind of triumphs and tribulations of pediatric cancer research from a translational clinical perspective. So this has been fascinating.
I wonder if I could say one more thing about funding this type of research.
Yes.
Because I see your yellow ribbon.
Yeah, I mean, right. So, you know, in terms of awareness, I mean, I think people, I get this all the time where people will make a donation to my group and they're like, oh, you know, I know it's not that much. It's only a few $1,000. And I can't emphasize enough how important that is. And I think part of it is people don't understand how research is funded. It's basically from three sources, donors who give directly to the lab, foundations, and then the National Institute of Health. Each of our institutions actually give us very little research to do, and very little money to do research, because so much of what their budget is operational, right? They need to make sure that the clinics are running, which is, of course, number one priority. So most of how we run our research groups, whether it's a clinical lab or a laboratory-based group, comes from those three sources. And the NIH is notoriously risk averse, right? So they'll only fund an idea that basically you've already proven is going to work. They're like a reimbursement agency almost. I don't want to disparage them because I'm lucky enough to have some NIH funding, and it's great. It's really great, and it really gets you to like get from like a pretty solid idea to total validation where you can maybe move it into the clinic. That part we're talking about. But you know, when ctDNA first started to come out, and I wanted to try it in a couple of patients who had Ewing sarcoma, where I happened to have some blood samples from our local biobank, the NIH wasn't going to fund that. And what it took was donor-directed funds. I had some donor-directed funds from a family that raised money because their child died from Ewing sarcoma, and I did a cheap old PCR, and we saw the presence of the Ewing mutation in a patient's blood sample. And it changed everything. It changed the entire course of my career, because now that's the main focus of my group. And without that donor-directed money, that would have never, ever happened. And so I think it's really important, and just it's really important right now, because all the foundations and the NIH will soon as well are going to kind of suffer around this economic downturn. And there are people who are not going to have the means to donate, and that's okay. But if you do have the means, don't forget that this is continuously a problem for us, is that kids are getting new cancers. We don't have good enough therapies. It's going to take us decades to figure out all the answers. Same with adult cancers, same with all the sarcomas. And so I think it's just really important that if you have the means to donate, it's really important to think about supporting your favorite cancer foundation right now, because they're actually hurting. They're even having to cut back on some of their grants, fund them at a lower rate than they had initially promised, or not release new grants for the upcoming academic year. And until we sort of turn things around, it's going to stay that way. So if you're out there and you're listening, you have the means to sort of double down or triple down on what you've done before, we would really appreciate it. And one thing I can promise is we will not waste it. We will use this to make advances in pediatric sarcomas, adult sarcomas. This is our passion. This is what we want to do, and we're not going to take any of those donations lightly.
Yeah, I would definitely echo that. You know, I had several foundation grants and several direct donors at my previous institution, and I think one thing that people don't know, I think, you know, whatever your political leanings are, you know, there is a certain amount of waste in state and federal systems, but these foundation grants and federal grants too, we are responsible for reporting what we're spending that money on. And a discrepancy of $1 will be caught. You know that because I've had some discrepancies down to like $10, and you know, they're like, you have an encumbrance, which is, you know, you have an outstanding bill that you haven't paid for your antibody conjugate or whatever, you got to reconcile that. So we are held, our feet are held to the fire to that. And that's important because that money is hard earned by somebody and then donated, and we have to be good stewards of that money. And I've always taken that very seriously. And I know all of my colleagues have too. And we are held accountable for it. So it doesn't, I feel like I can personally say, at least from my own experience, I'm sure you can vouch for this too, Brian and Lisa, is that it's not squandered.
That's right.
Right.
Yep.
And it's still very, very important. So
Yeah.
I agree wholeheartedly. September is Childhood Cancer Awareness Month.
Yes.
Yay.
Donate.
Well, we're over.
Yeah. I know. Well, we have to go, but I did want to mention, just because we did mention Dr. Painter, and that Brian works with her too, and there's an osteosarcoma project. So Brian, if you just want to talk about that for like 1 minute before we all sign off.
Yeah, it's a great podcast. Go back and listen to it. I think Corey and I work together well, and we've worked together on projects before.
She's amazing.
She's amazing, and together with, I mentioned Dr. Janeway before, together with her, we love her too. Yes, I'm sure she'll be a sarcoma sister on the podcast at some point.
We hope so.
We started an osteosarcoma arm, and that really has very similar goals to the angiosarcoma arm that's been so wildly successful at Count Me In. So if you have a loved one who is battling osteosarcoma, and wants to contribute to a research project that has really ambitious goals, look up Count Me In. The enrollment's online. It's very easy to participate. I do know that they just started kicking off getting samples again because everything was sort of locked down for a while due to COVID. So now they're starting to request samples. So everything is fully up and running with the Count Me In project again. And we'd love to get participation from hundreds of patients with osteosarcoma, because we need those numbers to understand everything about it, all the biology, and find new treatments.
Yeah, thanks.
All right, Lisa.
Yeah, well, thank you so much. Yeah, I know. Thanks. It's been so great having you.
Thanks for having me, guys. It's been a pleasure. I love your podcast. Keep it up. It's so important for our field.
We got one more listener, Lisa.
Yay! Thank you. Like us on the socials. All right.